on food microbiology. So in this part, first we review again the classification of microorganisms, how to classify them into different groups. Different groups have more or less different characteristics. They have different characteristics. So we divide them in groups so we know better how to control them or how to destroy them. And then we review a bit on microorganisms, especially on bacteria. And then we will discuss primary sources of microorganisms in food. From where do they come into food? Knowing this is important or not important? Important, so then we can prevent, we can limit them to contaminate into the food. We know from where they come, then we have measures or we have solutions to prevent as much as possible. And then the next parts we discuss intrinsic factors and extrinsic factors which affect the microbial growth. Intrinsic factors mean the factor belong to the food. For example, pH. For example, water activity of the food. Okay. Those are intrinsic factor affecting microbial growth. And we also discuss extrinsic factors. Extrinsic mean the factor belonging to the environment surrounding the food. Okay? Like temperatures, like relative humidity of the air surrounding the food. Why it is important to know these factors? Why? In order to have a longer cell life, we should promote or we should inhibit the growth of microorganisms during storage. Inhibit. So when we know the factors, we can modify the factors to inhibit them. So knowing this is very important. To inhibit them, for example, or to destroy them, and then you have a longer shelf life. You have less issue with food poisonings. And you know, for example, in your specific products, what would be the risks you have? What would be the risks of microorganism that you have? For example, if your product is dry, very dry, what could be the microorganism which can grow? If your product has a high water content, water activity, for example, then what would be? So that is during storage. But during fermentation, what should we do? We should inhibit or we should stimulate the growth of microorganisms. During fermentation, both. We stimulate the growth of the good microorganism, the starter coater, for example. But we should inhibit the growth of undesired microorganism. So that we should do during fermentation. And how to do this? If we know the extrinsic and intrinsic factors that influence the growth of these two groups, we can do this. We can stimulate one and we inhibit the other one. Okay, now we do go into detail. Classification of food microorganisms. The classification can be based on taxonomy, on the species and the genera and so on. Về loài, về phân loài. Then we can divide microorganism into mold, into yeast, bacteria, and viruses. Okay, you studied this already in the other course, food microbiology. Which one is the smallest among these four? Which one? Viruses. Viruses is the smallest living being. It's much smaller than bacteria. Viruses is obligatory parasite. What does that mean? Means that this virus cannot multiply without a hot cell. If you put a virus in a medium, for example, medium là môi trường dinh dưỡng cho vi sinh vật. If you put virus in medium, they will not multiply. They will not grow. They only grow in hot cells. tế bào chủ. They infect human cells, infect plant cells, then they will multiply. We talk a lot now about coronavirus, you know. Okay, when they are there on environment, they will not multiply in number. Only when they infect in human bodies, in the cells, then they will multiply, grow in number. Okay, but these three can multiply on themselves. If they have good conditions, if they have nutrients, then they will grow in number. In microorganisms, when we use the word grow, then means that it also means multiply in number. Trong vi sinh vật, cái từ phát triển nó không có nghĩa là trở nên to hơn mà là tăng lên về số lượng nha. So in microbiology, the grow means the increase in number, not the increase in size. In microbiology, the two words is the same, division and multiplication. Chỉ trong môn vi sinh vật, cái từ chia nó tương đồng với cái từ nhân. Hiểu chỗ này không? Có nghĩa là một tế bào nó làm sao? Nó phân chia ra. Mà thực ra nó là đang nhân lên số lượng. Okay. So in microbiology, the word division is similar or has the same meanings as multiplication. This is strange but true. They divide the cells, but they increase in number. Okay, so now we go back somewhere here. Mold is the largest in size in these four. They are multicellular and filamentous fungi. So mold and yeast has the common name, fungi. But the mold have many cells, multicellular. But the yeast have only one cell. And then bacteria, one cell, unicellular. The size of bacteria is somehow several micrometer. Can you see by your eyes with this size? No. The size of yeast is somehow like 10 times larger than bacteria. Can we see by our naked eyes? Yes or no? No. 40 micrometer. How can you see? Still very small. You cannot see. Mold. Can you see by your naked eyes? Yes. If you look at a bread with a lot of mold grow, you see them. It can be long like several centimeter already. That's mold. And then we have another way to classify food microorganism based on the influence to food and health of human. We then can divide microorganism into spoilage group, into starter coater and pathogenic microorganisms. Which group do we want them to grow during fermentation? This one. Which group do we want them to grow during storage of food products? We don't want them to grow during food storage. If this group grow during storage, what would be the result? Food gets spoiled and then the cell life is shorter. So do we want them to grow? No. In these three groups, this one is the most critical. We don't want them to grow because when they grow, the food sometimes look okay. Okay, means that the food do not look spoiled, but when you eat, you get sick, and then this is the reason for what? Food poisoning. Okay, why it is dangerous? Because they grow, the food look okay. And then we can classify the microorganism based on the tolerance to temperatures or the relation between the grow and temperatures. Then we will see in a while in other slide how to classify. Now we have some information